we'll be looking at 1, 2, 3, 4, 5, 6 coenzymes. Earlier on, we referred to coenzymes as components that are prosthetic parts of the enzymes, which mostly are proteins. So the non-protein part of the enzymes which are needed to make these enzymes active to carry out their functions. And we actually distinguish between coenzymes, cofactors, even though sometimes these terms are used interchangeably. We saw the cofactors to be metal ions, such as zinc ions, copper ions, magnesium ions, etc. But the coenzymes are some organic compounds, but they are not proteins that act together in complex with the apoprotein or the apoenzyme, the protein part of the enzyme, to carry out the function of the enzyme in catalyzing biochemical reactions. So we will first look at, so what we'll do is that when we take any of the coenzymes, we'll look at the structure of the coenzyme. So if we take thiamine pyrophosphate, the first one, we'll look at its structure, and then we'll move on to look at its function. We'll do same for the nicotinamide nucleotides, Flavin nucleotides, Coenzyme A, Biotin, and Folates. So that's what we are coming to do today. We just look at the structure, and then we continue to look at the function, the kind of reactions that they are involved in. So the first is the thiamine pyrophosphate. Now, in comparison with vitamin B1, which is thiamine, the difference you see in the structure of thiamine pyrophosphate and B1 is that if you look at the structure of vitamin B1, you see the 6-sided ring here. That same 6-sided ring, you will see it also in thiamine pyrophosphate as the coenzyme. In place of this CH2, you just have this side, this vertex here, which is showing that there's a CH2 there. This ring, the thiazolium ring, which you see here, is the same ring which we are seeing in thiamine pyrophosphate. CH2, CH2 is the same. There's a CH2 here, there's a CH2 there. So the only difference you see in the structure of thiamine and thiamine pyrophosphate is that in place of the hydroxyl group, this terminal hydroxyl group in vitamin B1, you have the 2 phosphate groups. So these 2 phosphate groups is what actually is being referred to as a pyrophosphate. A pyrophosphate is also called a diphosphate, so 2 phosphate groups. So in other words, you can say that vitamin B1 is a precursor for thiamine pyrophosphate, or the activated form of vitamin B1 in a biological system is the diphosphorylated form of the compound. So you can say as the vitamin is phosphorylated with 2 phosphate groups, it becomes active to act as an coenzyme in biological or in biochemical reactions, acting as a coenzyme to some specific enzymes. Now you see also that what we refer to as the thiazolium ring, this ring is having uh, some portion of it which is written in a different color, in magenta or say red. This carbon number 2, is red, and the hydrogen on it is also red. And that is intentional to show that it is this specific portion of the coenzyme which is reactive. It is that portion which is involved in its function as a coenzyme. Now the name thiazolium ring, to the fact that the ring contains sulfur and nitrogen. So I mean, in chemistry, when you see the term thio, Thio, or thia, which is Thia, it's referring to a sulfur component that is in a molecule, and azo refers to the nitrogen component in a compound. So that the two of them combined together is gives the name of this ring as thiazolium ring, which is the active part of the molecule, specifically the carbon 2 and the hydrogen that is on it. As you soon see in the function of this coenzyme, you see that the portion that is actually involved in its function as a coenzyme is that carbon number 2 and the hydrogen that is on it as the reactive part of the molecule. Now, here you see in the first column examples of enzymes to which thiamine pyrophosphate acts as a coenzyme. So the first enzyme is pyruvate decarboxylase. So from your earlier knowledge on enzymes, just by looking at the name, you know that this is an enzyme which is going to be acting on pyruvate as a substrate. And the activity it will have on pyruvate is to remove carboxylic acid group from it, from the second part of the name, decarboxylase. So it will remove a carboxylic acid group from pyruvate. Now, pyruvate is the final product of the glycolytic pathway. When you go back and look at the process of glycolysis, you realize the end product of that process is out of which you have acetyl-CoA, which then goes into the Krebs cycle or the T cycle. Now you see that one of the pathways, so a pathway in which pyruvate decarboxylase is in ethanol fermentation. Now, if you look at the bond, so the bond on which the enzyme will be acting on is that bond which is close to the carbonyl group, the carboxylic acid group. So it's between the carboxylic acid group and this C double bond O group that you see there. And that's, so you see that bond is actually in red. So all the bonds that the enzymes are acting on intentionally have been presented in red, different from the other colors, the other bonds which are in black. In this case, the enzyme is acting on the bond between these two carbons, the carbon carrying the carboxylic acid group, and then this carbon with the forming the C double bond O. Just a moment, I think. All right, so sorry about that. Now, you also see that the bond, which is between the C double bond O and the carboxylic acid group, uh, after it is broken, a new bond is formed. And that bond is also in red. So in other words, this group, the carboxylic acid group, which is broken off, of course, that is where the second part of the name of the enzyme, pyruvate decarboxylase. Decarboxylase is coming from. So this C double bond O is broken off. And after it's broken off, you now have in place of it this hydrogen. So a new bond is formed there. That's an aldehyde being formed there. So that in the fermentation of alcohols, you have this uh, process of reduction of carboxylic acids into aldehydes, which later can get oxidized into another carboxylic acid, or from here it gets further reduced into an alcohol. Now you have pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase. Now, when you look at this, the two names, there's something common about them. Dehydrogenase, dehydrogenase. So when you see the name of any enzyme, dehydrogenase, quickly you should know that that enzyme is an oxidoreductase. So you remember in our classification of enzymes, one of the first class that we saw was oxidoreductases, enzymes that are involved in oxidation and reduction reactions, what you commonly refer to as redox reactions. One which is broken here is similar to what you have there. So it's still pyruvate, and it is still that same bond that is between the carboxylic acid group and the C double bond O, which is broken. But in place of the hydrogen which binds in the first, bond formation that we saw, in place of that, you now have the conjugation of coenzyme A. So you have this coenzyme A. So that is, you see this reaction in the synthesis of acetyl-CoA. So that it means that after you have the formation of pyruvate from glycolysis, you have this process of formation of acetyl-CoA, which then goes into the citric acid cycle or the Krebs cycle. So that is what you see here. Under pathways, you see it's here as synthesis of acetyl-CoA and then the citric acid cycle. So you have the acetyl-CoA going into the citric acid cycle. Then also you can have it in one of the pathways of metabolism of carbohydrates, which is the pentose phosphate pathway, which goes into the synthesis of nucleic acids. So you will see these things much more in detail when we are looking at the metabolic pathways of the biomolecules that I mentioned earlier at the beginning of the class. So that if you have an enzyme transketolase to which you have the coenzyme thiamine pyrophosphate acting, in this case, you see that even though the group that is being removed from this compound, which because the bond which is broken is between these two carbons, that is the carbon carrying the double bond, and then the carbon that binds to it, you have that bond broken. The new group which can, even though they look alike, you see this is intentionally the side of the compound is labeled as R, and this is R5, meaning that the continuation of the compound, what you have here, that side chain, that residue is different from what was on the original compound, which was broken. So even though they look the same, they are not the same, because you can have the residue that is attached to that carbon carrying a hydroxyl group being different. So you can say the transketolase is breaking the bond which is between this ketone group and the carbon with the hydroxyl group on it, specifically on that bond. That's what it is. It is acting. That's what the enzyme is acting on. And the others you see also again, C double bond O, C double bond O in all of them. Attached to it, you have some carbonyl group again, which is the carboxylic acid group. So those are specific bonds on which the enzyme will be acting, and the coenzyme thiamine pyrophosphate will be aiding in that process. Right. Now, looking much into the mechanism of action of thiamine pyrophosphate, that is how the coenzyme acts in the process of biochemical reactions, in which it is acting as a prosthetic part of the enzymes that are involved in those processes. So if you are looking at, so what you see here actually is a cycle of a reaction in which thiamine pyrophosphate is involved. So looking at it clockwise, I mean, you have reaction 1, reaction 2, reaction 4, which means we are going clockwise from the right top down, then we go left, and then go up to the, and then the cycle continues. Now, I must warn that you shouldn't, say you are chewing this thing in your mind. Just try to understand it, like what thiamine pyrophosphate is doing as an coenzyme, but not to be chewing it. Look at it reaction by reaction and see what you notice, what really is happening in this process. So you can look at the coenzyme like a platform on which a substrate is stabilized. Yeah, I mean, if you can make an analogy, like you are mentioning you want to chop some vegetables, and you need a chopping board to place your vegetable on it to be stable before you take your a knife to chop that vegetable on it. So the coenzyme is serving as that chopping board, stabilizing the vegetable. So the coenzyme, in this case, thiamine pyrophosphate, is acting as that platform which is stabilizing the substrate upon which the enzyme should be acting. So that substrate is pyruvate. So you see pyruvate, which is in a different color, red, and you see that the when we were looking at the structure of the thiamine pyrophosphate, we refer to the thiazolum ring as that active part of the molecule, and specifically carbon number 2 that has on it, which was in red, and we refer to that portion as the reactive part of the molecule. So you see it here, that reactive carbon loses the hydrogen that is on it. It donates, it gives out that hydrogen that is on it, and it is now a carbanion. It's just an anion, which means that you're having some lone pair of electrons on it, and it's having some nucleophilic attack on that carbon, which is carrying the double bond O. So it's that ketone group carbon. Now, I mean, what you actually see is that there is some temporary formation of a covalent bond with that carbon carrying the C double bond O on the carbon which is reactive in the thiazolum ring. So you see, so you can look at it as if that carbon number 2 is the seat. That is the seat on which the substrate now is sitting. So it's stabilized in that manner. Now you notice that the C double bond O, that double bond O which is on it, is now reduced to a hydroxyl group. So you see there is some hydrogen ion which comes into that reaction and reduces that double bond O to a hydroxyl group. And then you notice that, you know, the enzyme pyruvate decarboxylase, I said from the beginning, the name tells you that it removes a carboxylic acid group from the substrate pyruvate. So you see this carboxyl group, the C double bond OO, C. So that is what comes out as CO2. Then you are now left with the residue, which is having just two structures forming in that process. So now you see that after there is that stabilization of the molecule, there is that temporary formation of a covalent bond with the thiazolum ring. The enzyme then acts in removing the carboxylic acid group. And then you see the reaction continues such that you now have the formation of the acetaldehyde. Because what you saw from the table earlier on, in the end, you are having the formation of the bond with hydrogen replacing the carboxylic acid group, which is removed from it. So from here, when you have this carbon ion being formed, this OH group is oxidized as the hydrogen ion, a proton, comes into the reaction and gets attached to that carbon to give you the aldehyde group, so that the product of this whole process becomes acetaldehyde. So you have the removal of carboxylic acid group, and then in the end, you are having some oxidation reaction because of the change of the hydroxyl group to the C double bond O, and at the same time, some proton or hydrogen ion coming in to bonded to that carbon from which the carboxylic acid group was broken to now have your acetaldehyde. And then you have that dissociating from the coenzyme. The coenzyme is free to go back into that cycle of the reaction. So, like I said from the beginning, I warned that it's not something it should be. You shouldn't go cramping this in your head like you are chewing it, that sort of thing. But you should just look at, especially when you have these things written in different colors, like the compound substrate which the enzyme should act on is in a different color, and you see at least some formation of the bond with a carbon which is reactive in the coenzyme. Those are the things you should be looking at and say that, okay, the substrate is actually having interaction with the coenzyme, stabilized on it for the enzyme to act on it. And the specific action of the enzyme is to remove the carboxylic acid group. So you look at this carboxylic acid group, which is here, and you now see that it's coming out, is being released as carbon dioxide, and then you are left with a residue which further undergoes some oxidation reduction reactions and to form the product acetaldehyde. All right. Now the next coenzyme is nicotinamide nucleotides. Now these two you have seen these coenzymes in many biochemical reactions, but maybe you didn't pay so much of attention to them because you were just looking at the reactions in terms of the reactants and the products. And so whatever else was happening in the middle, you may not pay so much of attention to it, but you will see these nucleotides a lot of times being represented in chemical reactions. Now, the nicotinamide nucleotides, when you look at it, you realize that it's coming from nicotinic acid, which is vitamin B3. So take note that thiamine pyrophosphate is vitamin B1, which is just a conjugation of two phosphate groups, which makes it a coenzyme, thiamine pyrophosphate. Now, as we have moved on to the nicotinamide nucleotides, again, this is a derivative of a vitamin B. This time around, it is vitamin B3. And vitamin B3 is niacin or nicotinic acid. So you see the difference between nicotinic acid and nicotinamide is just that the carboxylic acid group. So if this carboxylic acid group was written in full, this oxygen with the negative charge on it would have been a hydroxyl group. So it means that in place of this hydroxyl group, you have an amine group, making it nicotinamide. So what you see is that you have two nucleotides that are fused together. So the first nucleotide you will notice is this adenosine. So you see that is a 5-carbon sugar ribose. And on carbon 1, you have adenine, a nitrogenous base attached on that ribose. And there is a phosphate group on carbon number 5. Then the second nucleotide you see is this one with 5-carbon sugar, again, ribose. But this time around, you have nicotinamide being bonded on carbon number 1. You know, there's also a phosphate group on carbon number 5. So these are two nucleotides that are bonded together. So if you remember way back in your first year biochemistry, you studied nucleotides being the basic unit of nucleic acids, which are made up of a 5-carbon sugar. If it was in RNA, it would be ribose. If it was in DNA, it would be deoxyribose. And on carbon number 1 of these 5-carbon sugar, you have the nitrogenous base. So you see the nitrogenous bases on the carbon number 1. And then on carbon number 5, you have phosphate groups. That's which you see, the phosphate group here. Except that the structure of this, the difference that you see, one difference you see with the bonding of nucleotides in nucleic acids is that you see when you look at the structure of DNA, RNA, and you are talking about the phosphodiester bonds that are formed between the nucleotides, you have that phosphodiester bond forming between carbon number 5 and carbon number 3. So remember that, I mean, when you were doing nucleic acids in first year biochemistry, you talked of the 3' end of the nucleic acid and the 5' end of the nucleic acid. And that's because of the fact that you are having the phosphodiester bond that is between the nucleotides in a nucleic acid chain, forming between the carbon number 5 of the nucleotides and to the next is to be on number 3. So what I mean is this phosphodiester bond, which you are seeing here, these are that are bonded together. This ester bond, you see, is being formed between carbon number 5 and another carbon number 5 of the next nucleotide. If it was in nucleic acid, you see, this bond will have rather been carbon number 3. So in place of this hydroxyl group, you would have been seeing the bonding of this second phosphate group there, if it was in a nucleic acid. That's one major difference you see with the nicotinamide, the structure of nicotinamide nucleotides the sort of the phosphodiester bond forming between them when you are comparing them to the phosphodiester bond which is forming between nucleotides in nucleic acids. So take note again, this is vitamin B3. Thiamine pyrophosphate is coming from vitamin B1 because I will make reference to that fact, getting to the end why that you are having, different forms of vitamins that are serving as coenzymes. So it was just phosphorylation of it, two phosphate groups attached to that, making it active to be acting as a coenzyme. In this case, this vitamin B3 is getting attached to a sugar and phosphate group to make it a nucleotide, and at the same time you have a separate nucleotide which is the adenosine bonded to it. So in this case you have both phosphate groups and nucleotides binding to that vitamin, making it a dinucleotide, and so that becomes an active coenzyme. Now, take note also that on adenosine, usually you refer to this nicotinamide nucleotide as NAD. Sometimes you see the oxidized form as NAD plus and the reduced form as NADH. And you also see the phosphorylated form, which is NADP. So you can see NADP plus or NADPH. Now, that phosphate group, it binds to the second carbon on adenosine. So in place of this hydroxyl group on adenosine, if you have a phosphate group there, that will give you NADP. Okay, we can move on and look at the function of the nicotinamide nucleotides. Now, also just like the TPP, but this specifically is with oxidative reductive reactions. And what this coenzyme does is to transfer electrons, together with protons from one molecule to the other. So remember, oxidation reduction reactions occur simultaneously because anytime there's oxidation occurring, some other compound gets reduced. And if there is reduction occurring, the second compound that gets oxidized. And in this case, you have the nicotinamide nucleotides carrying electrons and protons in this process. And there is some specificity to this, the specificity of the coenzymes, where actually, so you see that the coenzyme is actually being referred to the nicotinamide ring, which is in red here. You see it being presented as A side and B side. And what it means is that if you have the protons or these hydrides, the hydrogen ions being transferred from one molecule to the other, if that hydrogen binds to, if you are looking at the stereochemical structure of the molecule, that is that the molecule in a 3D form, that molecule will have the part which is facing you and a molecule, then the part which is behind it, so the part which is away from you. So if you are looking at your screen, the acid, the nicotinamide group, you have, if it's a 3D in front of you, the front of it is what you see, and you don't see the back of it. Now, if it happens that the hydrogen which comes binds to the front of it, the front is what is being referred to as the A side. So that blue hydrogen comes to bind to the front, which is facing you, that you can see. But it can also be that the hydrogen goes to the back of that molecule, which means that compound, the nicotinamide, is turned the other way from you, so that the hydrogen binds to the back of it. That's what is being referred to as the B side. So we'll see further how that specificity works with different enzymes. Now, the nicotinamide nucleotides in this transfer, they actually take the hydrogen ions electrons and move to another molecule, transfer it to the molecule. That is how they act as electron carriers in oxidative reduction reactions. Now, in that process of oxidative reductive reactions, which generally occurs in, say, the mitochondrial matrix, or sometimes maybe in the cytoplasm. We saw earlier that you can have the oxidized form of the nicotinamide nucleotide, which is written as NAD+, and then the reduced form, which is NADH. And we also saw that if you have on carbon number 2, a phosphate group attached on that adenosine, then you will have NADP, and the reduced form of it will be NADPH. Generally, having oxidative reduction reactions in which you are having these nicotinamide nucleotides acting as coenzymes. You have NAD in higher quantities to be acting in oxidative reactions than NADP. So, I mean, what I'm talking about is that you have NAD+, that is in an oxidized form. That if it receives electrons from a molecule, it will become reduced, which means that molecule then becomes oxidized. The same way, if you have NADP, when NADP receives electrons from a molecule, or it dries from a molecule, it becomes reduced, whilst the molecule becomes oxidized. So in that case, you can have NAD+ and NADP, these two acting in oxidative reactions, that is causing a molecule to become oxidized. Then you can also have NADH and NADP, actually will be the ones that donate either the hydride ions or call it electrons, because the electrons move together with the hydride ions to the molecule, which means they will cause a reduction of the molecule, whilst they themselves become oxidized. So if you are taking NAD+ and NADP, these two, which can oxidize molecules, one of them, which is the NAD+, is in much higher quantities in biological system than the NADP. So it is the NAD+, which is prominent in oxidative reactions. And in a similar manner, when you come to NADH and NADPH, the NADP is prominent in reduction reactions than NADH. So that is what is being depicted in this third point, that NAD+ generally functions in oxidations, and NADPH is the usual coenzyme. Now, this is a specific example of a biochemical reaction in which you have the nicotinamide nucleotide acting. So this is, say, the metabolism of alcohol. So you see that is ethanol, and that ethanol gets oxidized to give you acetaldehyde as a first reaction. The acetaldehyde can get further oxidized to give you acetic acid. And the enzyme which is acting in that process is alcohol dehydrogenase. Then you see the coenzyme, there is NAD+, and it means that is what is accepting electrons and ions to be NADH. So itself becomes reduced. And then the substrate, which is the alcohol, becomes oxidized. In some instances, this two-step reaction, the first enzyme is called alcohol dehydrogenase, whilst the second enzyme is referred to as acetaldehyde dehydrogenase, because the substrate on which the enzyme is acting in the second step is acetaldehyde. So that it will not be alcohol dehydrogenase, alcohol dehydrogenase. When you are reading, you see alcohol dehydrogenase 1, alcohol dehydrogenase 2, but it's referring to the same things in different ways. Right, so I mean, it is a common metabolism reactions which you come across in biochemistry. How ethanol gets metabolized in the body, first into acetaldehyde, and then secondly into acetic acid. So to digress a bit, you see that in the first reaction, acetaldehyde, which is formed, can have some adverse effect on the nervous system. It is actually what causes things like the turning of the head, headaches, and even people losing their balance when they take in too much of alcohol. But if the alcohol is not that, it means that acetaldehyde is not going to be accumulating in the system to be giving that adverse effect, because it will quickly get oxidized to the acetic acid, and the acetic acid can easily get conjugated with coenzyme A, and that coenzyme A giving you acetyl-CoA, which then goes into the TCA cycle, and thus it will just generate energy with some release of carbon dioxide from the body as a waste product. All right. So this is very much on the stereospecificity that I was talking about much earlier concerning the amide ring, which is having an A side that accepts the hydride ions and electrons from the substrate that the enzyme is acting on. And what you see here, the table you see here, is referring to the coenzyme acting on specific enzymes with some specificities, the stereospecificity. So for instance, what it means is that isoside dehydrogenase having coenzyme NAD+, you have that coenzyme accepting hydride ions on the A side. So it means that the coenzyme NAD will have that specific configuration of having the A side to receive the hydride ions from isocitrate reaction to occur. So that if NAD+ was in a configuration with a B side close to isocitrate, that reaction will not occur. So it's a form of a biological switch on and off. If it turns A to the side of the substrate, the reaction occurs. But if it turns the backside B, the reaction will not occur. And you have that, so alpha ketoglutarate dehydrogenase we'll be having the coenzyme with a configuration of the B side that is acting with that enzyme. And that is what you see with other enzymes as well. You see that as stereospecificity with different enzymes. The example which we just saw, alcohol dehydrogenase, NAD plus, will be in the configuration of having the A side, accepting the hydride ions from alcohol. So you can look at the see whether it is A side or B side. So these two is not something that cramp in your heads. It is for you to have an understanding of how you can have stereospecificity given the function of these coenzymes as biological switches. Then the next is Flavin nucleotides. Flavin nucleotides. So the nucleotides are also derivatives of vitamin B2, which is riboflavin. So if you take the molecule that 3 rings fused up to is CH2. So if in place of this phosphate group, you just have a hydroxyl group here, that will be structure of riboflavin, that is vitamin B2. So here again, you see there is that aggregation of the vitamin B2 with a nucleotide, which is again adenosine. So you see, and then there is a second phosphate group that you have this phosphodiester bond again forming the adenosine nucleotide with the vitamin riboflavin to give you the full structure of the flavin nucleotide. Now you can have flavin mononucleotide, which is just from these the 3 fused rings up here, the isoalloxacin ring with these polyhydroxy groups with 1 phosphate group on it. That gives you a flavin mononucleotide. But the whole structure, because that is a nucleotide, because this ring also you can refer to as a nitrogenous base, because you can see some nitrogen atoms in this fused ring structure, the isoalloxacin ring. And you also you have a second nucleotide. So if they come together, that's a dinucleotide. So the whole structure is a flavin adenosine or flavin adenine dinucleotide. Now you see this flavin nucleotides also involved in oxidative reduction reactions. As you can clearly see, there is some hydride and electrons being received by having adenosine or flavin adenine dinucleotide. It can receive, it can accept 1 or it can accept 2. So the first one, if it accepts that, you refer to that as a semiquinone. What is formed at the end of it is a semiquinone. If it receives 2 is the maximum we could receive. If it receives 2 and become FADH2, that is now the flavin nucleotide becomes fully reduced. So you see that's the first hydrogen and then the second hydrogen in there. That is a fully reduced flavin nucleotide. This is a semi-reduced flavin nucleotide, which is referred to as a semiquinone. And you see these flavin nucleotides, acting also in oxidative reduction reactions. You can say that the active part of this coenzyme is the isoalloxacin ring. Of course, it is on that isoalexaxin ring that you see the hydrogen ions being accepted. So you see the first one being accepted on this nitrogen of the middle ring, and then this other free nitrogen off the last ring. You can see the two hydrogens being accepted there. So which means it is this ring, which is the active part of the coenzyme. Now, you see the activity of these Flavin nucleotides, such that you can have some enzymes that are having the dinucleotide as the coenzyme, and some of them will have the mononucleotide as the coenzyme. So you see in this table that some of these enzymes, like acyl-CoA dehydrogenase, is having the dinucleotide FAD. So all these other enzymes here, up to thioredoxin reductase, they are all having nucleotide as the coenzyme. Then class 2, which is the NADH, and then the glycolate oxidase, they are having more. Now you have one major difference between the Flavin nucleotide, the nicotinamide nucleotides, being that the nicotinamide nucleotides, they actually move between one molecule to the other, carrying the hydride ions and the electrons. But the Flavin nucleotides, they bind tightly to the enzymes to which they are acting as coenzymes. Because of that, we refer to those enzymes as flavoproteins or flavoenzymes. They are actually bound tightly together. And you find them acting as these flavoproteins. You see them acting like receptors of light, specifically being referred to as cryptochromes. So these cryptochromes, they mediate the effect of light on plant development. You pay attention to plants, plant behavior at sunrise, sunset. Many of these plants is very predominantly, you can see that in the mornings when the sun is rising, these plants will have their leaves opening, or their flowers, if it's a flowering plant, flowers opening. And in the evenings, you see that the leaves are closing. Or if it's a flowering plant, you see the flowers are opening. This is where you see of these flavoproteins acting in what to as a biological clock in these organisms. In mammals like us, we also have such biological clock, which we refer to as the circadian rhythm. We react to it with some participation of the endocrine system, where you have the pineal gland, somewhere below the hypothalamus, secreting a hormone, melatonin, which regulates our sleep pattern, so that when it is in the night, the pineal gland begins to secrete this hormone, melatonin, which induces sleep, and it is an amount of entering our eyes. So the light that is entering our eyes is actually regulated. If it is dark, it regulates the secretion of melatonin, so that there will be increase in the secretion of melatonin to induce sleep. And when it is sunrise, the sun is rising, there is a reduction of that secretion of the hormone melatonin, so that we wake up. So you have the rhythm in us. Now you can also have these flavoproteins also acting as lyases. So the classes of enzymes of lyases, which were involved in either the removal of groups or addition of groups in molecules. if it is a removal of some molecule to form double bonds, then it is to remove the double bonds from this from this substrate. We saw that class of enzymes, the lyases. So there's a group of these lyases which we refer to as the photolyases, which are found in bacteria or generally some prokaryotes, even some eukaryotes, which is involved in the have in the structure of DNA, for instance, you can have these flavoproteins with activation from light in this process of repair of these nucleic acids. Then we have the coenzyme A. So the coenzyme A is one of the commonest coenzymes seen over and over again, involved in so many biochemical reactions. And you see, this is a complex structure of that enzyme A. Every time that's CoA. Actually, this is the whole structure of, again, here. Think this part, the shaded part, the pink shaded part, which is pantothenic acid, is also one of the vitamin Bs. I think that is vitamin B5. So again, you have this vitamin B5, which is conjugated to so again, you see adenine is there, and you have, so this is a whole adenosine, except that on carbon number 3, you have another phosphate group. So that is phosphoadenosine. So you see the name is 3' phosphoadenosine diphosphate. So 3' meaning the phosphate group is on carbon number 3. It's a diphosphate because there are 2 on carbon number 5. And you have that phosphonucleotide, or call it a nucleotide, is just for carbon. And then another residue, which is the captoethylamine. Now, intentionally, the very last end of the molecule, a sulfur hydral group, is written in red because that is actually the reactive part of this whole molecule. So this whole large molecule coenzyme A, the part of it which is reactive is this sulfur hydral group, which is also referred as a thiol group because of the sulfur that is in there. The sulfur hydral group is that reactive part. So for instance, what you write as acetyl-CoA, this is the acetic group, the acetyl group on which this whole molecule, the coenzyme A, is binding, so that when this hydride is given out, is released, is donated out by this whole molecule, the sulfur then binds onto that acetyl group, and the of this part is what you write. Times you see it being written as just the bond and CoA without the in many reactions. So for instance, even in glycolysis, where after the formation of pyruvates, from pyruvate, we saw in this lecture where you have pyruvates after decarboxylation, you have the acetyl group binding to coenzyme A. In one of those, I think 2 different reactions that we saw, that there was the synthesis of coenzyme A to go into the TCA cycle. Now, the other one is biotin. Again, this is vitamin B. about vitamin Bs, different vitamin Bs. So you can see that flowing through. Now, you see the type of reaction that biotin is involved in here. I mean, if you look at, that's a bicarbonate. Again, a pyruvate, which is the end product of glycolysis, and the formation of oxaloacetate. I mean, this is one of the intermediates that you see in the TCA cycle. When you look at the first reaction, when the acetyl-CoA enters the carboxylic acid cycle, it actually it actually binds with the oxaloacetate to give you isocitrate. So this is again in carbohydrate metabolism that you will see this. Now the structure of the biotin, again, 2 fused rings, and then you have some side chain of carboxylic acid, valeric acid attached to that 2 fused rings to give you the whole structure of biotin as the vitamin B7. Now, one of the things about vitamin B7 is that the consumption of, so see the consumption of raw eggs, for instance, contains the protein avidin, and avidin actually reduces the permeability, or say the absorption of the vitamin B7, which is biotin, into the system, so that the consumption of raw eggs can actually cause deficiency in vitamin B7, just because it goes to prevent the absorption of the vitamin into the bloodstream to be available to the biological system to use. And that's over a long period of time, it can result into the thinning of hair, fingernails being weak, can cause some skin rashes. Now, we'll soon see the sort of reaction, the sort of function that biotin has in biochemical reactions. And generally, it is about carrying carboxylic acid groups from one molecule to the other. So you can see clearly from here that from this bicarbonate, this carboxylic acid group, which is from the bicarbonate, the red portion of the molecule, gets conjugated onto pyruvate here. So in place, this is CH3, in place of one of the hydrogens, you now have CH2, and then you have this group, the carboxylic acid group, getting conjugated there to give you the structure of oxaloacetate. So biotin is involved in the transfer of the carboxylates or carboxylic acid groups over there. So you see it clearly here that, so this is biotin, this is the biotin, and then if you take this round portion as the enzyme that biotin is acting as a coenzyme to. Now that enzyme, which is a protein, is having some amino acids in it, and one of the amino acids in those proteins is lysine. So if this is the lysine arm, that's so the chain of lysine. So if you go to look at the structure of lysine, you see that it's having an extra on it, and you see that you are having conjugation of that protein onto the coenzyme this. And it's going to accept the carboxylic acid group from these bicarbonate ions. Of course, there is some expenditure of energy, ATP, in that process. And you see the carboxylic acid group or the carboxylate coming from the bicarbonate is attached onto the nitrogen. So this nitrogen here, you now have the carboxylate group getting conjugated onto it. So that is how biotin carries the carboxylate group. And that carboxylate group now goes to get attached onto acetyl-CoA. So this is acetyl-CoA. So imagine this is acetyl-CoA, which is coming from the Krebs cycle, and you have the carboxyl group coming to get conjugated with it to give you malonyl-CoA. I think this is one of those reactions you see in protein metabolism. I think you will see malonyl-CoA being produced in that process. Now, we can, I think one of the things you can also draw attention to, interesting here, is that, when you into the lungs, and, we usually say that oxygen is taken into the stream, like the hemoglobin to the tissues. And then carbon dioxide is transported from the tissues back to the lungs to be excreted out of the body. Now, one interesting thing is that process which is described, that respiration process, is such that, in fact, only 15% of the carbon dioxide which is produced in cellular metabolism is excreted directly that way by the hemoglobin molecule carrying the carbon dioxide on it back to the lungs to be breathed out, exhaled out of the system. So it means that it is left with 15%, it's left with 85% of the carbon dioxide which is produced in that cellular metabolism. Now that 85%, some of it, that's 85%, you actually gets converted into carbonic acid, because it will react with water and form carbonic acid, and then after that it will dissociate into these bicarbonate ions and some hydrogen ions. When that happens, you can have hemoglobin also carrying the hydrogen ions back to the lungs, and you have the bicarbonate ions free flowing in the bloodstream. And when these free flowing bicarbonates in the bloodstream comes back to the lungs, and the hydrogen ions also carried by the hemoglobin to the lungs, the reverse of the reaction occurs, where you have the hydrogen ions, reacting with these bicarbonate ions to produce carbonic acid, which then dissociates into carbon dioxide and water. So we breathe out carbon dioxide with some water vapor. But you can also see that the byproducts, that carbon dioxide, which part of it is forming bicarbonate ions, is having some function also here. Another, it can have another pathway where you have it, being carried by this coenzyme biotin, and can get conjugated onto acetyl-CoA to form. That is a different metabolic pathway altogether. So you can see that there is an interconnection of these reactions in the biological systems. Right, I think this should be the last one, which is folates, which generally we refer to as folic acids. And here too, you see again, vitamin B9. So you see vitamin B9, which is being referred to as folic acid or just folates, and is being conjugated to amino acid, which is so in place of the nucleotides we were seeing, now you see that there is some conjugation with some amino acid. That's complex with an amino acid, and then you have some heterocyclic structure also attached on the other end of that folic acid. So again, you see that the precursor of this coenzyme is a vitamin B, and the active parts of the hormone, these nitrogens, nitrogen N5 and nitrogen N10, which interact with in the biochemical reactions, accepting hydrides in the biochemical reactions in which they are acting as coenzymes to some enzymes. Now you can relate to the fact that the folate or the folic acid is one of those common pharmaceutical products that are given to pregnant women, and that is because this coenzyme is very much needed in processes of fetal development, and specifically to the development of what we refer to as the neural tubes. In other words, it is the development of the nervous system, or you can narrow it down to say the development of the brain. So that's it. It's a very much needed vitamin for the development of the fetus. That is why in pregnancy it is very much prescribed for pregnant women to take, because the lack of it or the deficiency of it can actually affect the brain development or the nervous system development of the fetus. Now, this coenzyme is involved very much in the transfer of one carbon residues. So one carbon residue such as a methyl group like this, CH3, or maybe just an methylene group, CH2, a formyl group. This is sort of an aldehyde group, C double bond OH. So all these groups are just one carbon groups. And it is involved in the transfer of these groups in different metabolic reactions, which generally are referred to as folate-mediated one carbon metabolic pathways or metabolic reactions. So that is to do with folates. I think this should be the last coenzyme for us to deal with. Okay, this is just a summary of everything which we have gone over, which when I share the slides with you after going over it, you can look at it and see, just to have a summary of everything which we have gone through as far as the coenzymes are concerned. All right, so I think now I'll open the floor and listen to you. If you have some information to share with us, some questions, some comments, something to share with us. I think some of you perhaps have been working in either some hospital or some pharmacy, and so you have some experiences which you can share with us. So the floor is open now.